Hello, welcome to the session of plant protist. So in today's sessions, we will be discussing about the plant protist. But first of all, let's discuss that what are the general features of the protist, right? The moment we talk about the protist, so basically protists are, very well, that is a unicellular eukaryotic organizations. So primarily, they don't have any tissue grade of organization. That is the most important thing. The cell structure is complex and the cell wall is made up of cellulose. And they are known for very, very special locomotory organs. For instance, they have a pseudopodia, that is in slime mold, flagella in dinoflagellates, and cilia as well. The flagella and cilia have a typical eukaryotic structure that is 9+2 pattern. Okay. Nutrition, it could be photosynthetic, it could be holozoic, or even it could be saprobic. Okay. And some protists are mixotropic, as we observed in case of Euglena. Okay. Reproduction can be asexual as well as the sexuals, and we see there are two types of life cycle, and that is haplontic and diplontic life cycles, right? So first of all, let's talk about what exactly is a haplontic and diplontic life cycle. The three things what we have to remember in haplontic life cycle, that first of all, the dominant phase is always haploid, right? And here, the reductional division is going to occur after fertilization. For instance, you can see there, this is a haploid male, right? This is a haploid female plant. So both are going to form gametes. They will form zygote. And this zygote will undergo zygotic meiosis, and then haploid structure is actually being formed. Okay? So this is characterized by zygotic meiosis. And the typical example is dinoflagellates. But if you talk about diplontic life cycle, here what we see, the dominant phase is a diploid. They undergo gametic meiosis and haploid gametes are formed and they combine together to form zygote. So these are characterized by the gametic meiosis. So here, the reductional division is going to occur before fertilizations, as we observed in case of diatoms. Okay. So let's talk about first of all dinoflagellates. These are a group of the golden brown algae, right? And they constitute the main portion of the phytoplanktons. Classification wise, they belong to Dinophyceae of the division Pyrophyta. They are unicellular, motile and biflagellates. Most of them are found in the seawater, but some forms may occur in freshwater. The most important thing in case of dinoflagellate is the occurrence of the red tide phenomena, as you can see in the diagram as well. Okay. And there are two species well known to showing the phenomena of red tide, and that is the Gymnodium and Gonialax. Okay. In addition to that, they also show the phenomena of night phosphorus. But before that, let's talk about the cell structure. The cells are covered by rigid coat that are arranged in a plate-like structure. So they have a cellulose as well as the pectin, right? So the theca contains two groups, one what we call longitudinal group. Yes, you can see in the diagram, this is the longitudinal group, and then there is a transverse group, right? The transverse group is known as cingulum, whereas longitudinal group is known as sulcus, and each group is going to have one flagellates, right? So that is why we are calling them dinoflagellates. The pigments in the form of chlorophyll A, chlorophyll C, alpha carotene, and some abundant xanthophyll as well, and the food reserve is in the form of oils. The nucleus is very large, right? And one of the most important thing is that they have a nucleus called mesokaryon. So that is in between the prokaryote and the mesokaryote. So they have an organized nucleus, right? But the chromosome is not organized. So in the chromosome, the histone proteins are not there, right? So that's why we are calling them mesokaryon. And that is the most important thing in case of the dinoflagellates. As I told you, that some of them is going to show bioluminescence, and that bioluminescence is going to occur due to a protein called luciferin, right? And the species that is going to show bioluminescence, what we call Noculica, Gonialax, Pyrodinium. Okay, you can see in the diagram. So these are the bioluminescence what you can see over there, right? Now, some of them secretes a toxic material called saxitoxin, and they cause disease among the fishes, and that disease is actually called as a paralytic cell poisoning disease or PSP disease. It reproduces by asexual, and the sexual reproduction life cycle is haplontic, and the meiosis is zygotic. We have already discuss about in the haplontic life cycle, right? So now let's talk about the diatoms. So these are the golden brown algae, and as far as their classification is concerned, they belong to Basilorophyceae of phylum Chrysophyta. They are aquatic as well as the terrestrial, and one of the most important part is that they are good pollution indicator. Okay. Now the body is covered by the transparent siliceous cell having a silica. Again, there is a two valve, the outer called epitheca or larger called epitheca, and the smaller one what we call hypotheca. As you can see in the diagram, this larger one is actually called as a epitheca and the smaller one is called as a hypotheca. Okay. Now, as far as the pigment is concerned, pigments are in the form of chlorophyll A, chlorophyll C, and mind it is a beta carotene over there. They have a large number of fucoxanthin as well, and food reserve, since it is a chlorophyll C, so wherever you find chlorophyll C, the food is always going to be reserved in the form of oils, leucocin, and volatile. Commonly, it reproduces by binary fission, right? But what happens as a result of division, the size of one cell is actually decreases, okay, and that size is going to restored by a formation of rejuvenant cell, what we call oxospore. So the very formation of oxospore is very common. That is occurred in case of diatoms, right? So the plant body is diploid. So life cycle is diplontic. So it is a meiosis is going to be gametic meiosis. So the plant itself seems to be non-motile, but the male gametes that is formed by diatoms are motile and they bears a single flagella. Okay. So whenever the fertilization occurs, fertilization will form zygote and zygote will grow in size and they will form a oxospore. Oxospores eventually form the new plant. The ecological significance of diatoms is that they are important part of the phytoplankton and they are a major source of food for aquatic animals like whales and dolphins, right? Now finally come to Euglena. You know, Euglena is a freshwater protist, right? And it is a free-living, unicellular, facultative autotroph. So basically they are heterotroph, but when light is there, they start behaving as an autotroph. Nutrition is mixotropic. That is, in mixotropic, what we can say, holophytic and saprobic. That can feed on dead and decaying substances as well. Okay. So pigments are in the form of chlorophyll A, chlorophyll B, beta carotene and xanthophyll, and food is stored in the form of paramelamin starch. Now let's talk about the character about the cell wall. The cell structure, you know, cell wall is absent. So body is covered by plasma membrane followed by a helically arranged strips called pellicle. Locomotion is flagellar as well as creeping movement, and creeping movement is known as metaboly, and that movement is occurred due to presence of myonin fiber. Asexual reproduction occurs by longitudinal binary fission. That is important. So there is going to have a longitudinal binary fission, right? And Euglena is often considered as a connecting link between plant and animal because they have a few characters similar to plant and characters similar to animal as well. So as far as animal-like character is concerned, the absence of cell wall, the presence of pellicle, the presence of myonin fiber, the presence of contractile vacuole, the holozoic feeding, these are, or presence of mouth, these are the animal-like feature. Okay. And if you talk about plant-like feature, the presence of chloroplast and autotrophic nutrition, and the Euglena chloroplast contain paranoids that is used in the synthesis of paramylon. So these are the plant-like character, right? So this is all what I have to discuss as far as the plant protists are concerned. Thank you.